It's a pleasure for me to present Dr. Jose Donato Jr. He is a full professor at the Department of Physiology and Biophysics Institute of Biomedical Science of the University of São Paulo, Brazil. He's coordinated the Laboratory of Neuroendocrinology and Metabolism. He holds a master's degree in the food science and PhD in morphofunctional science from the University of São Paulo. He was a postdoctoral fellowship at the University of Texas Southwestern Medical Center. He has a postdoc. Dr. Donato investigates how neural circuits that regulate energy and glucose homeostasis are affected the action of several hormones. He has published more than 170 papers, and he received several national and international prizes, including the Return Home Fellowship from IBRO, Early Investigator Award for the Endocrine Society, a Spear Young Investigator Research Award in Endocrinology from Pfizer, an Excellent Award and New Research Leadership for the University of São Paulo, and supervisor of the winning board on the University of São Paulo outside. He is a mentor for more than 20 master, PhD, and postdoctoral students, and it's a pleasure for stay. Welcome to.
Thank you. Thank you very much, Maricela, for this very nice introduction. I would like to thank you all for this amazing opportunity to be here. It's my first time in Mexico. I always want to come to this country. So when we were in Dallas for 4 years, we always discussed the possibility to spend some trip here, and finally we were able to manage it and come to these jornadas académicas. Yo no hablo muy bien español, portuñol. That's why we are going to speak in English, but anyone that wants to speak to me later, it's going to be a pleasure. We can speak in English or try a portuñol. That would be extremely pleasant. So I will talk about the central actions of growth hormone. And one of the amazing things to visit the institute here is to know that the other people are also interested to understand the role of this hormone in the brain. So in the previous talk, we had a little bit about the information about this hormone, this growth hormone. We know that it's produced from the pituitary gland. There is also extra-pituitary production. The growth hormone, it's a powerful inducer of cell proliferation, tissue growth. It's one of the major factors that made us growth in terms of size. Of course, it's not the only one, but if you don't have growth hormone secretion, you will have a dwarfism. If you have it too much, you will have a gigantism. So it's an extremely important hormone to regulate our tissue growth. However, I would like to highlight other functions of this hormone today. And one of the functions that sometimes we forget, but because we always pay attention to the name, it's a hormone that makes the people grow, the animals grow. But growth hormone has important metabolic effects too. The classical effects described really early when this hormone was discovered were an increased gluconeogenesis in the liver, so increases endogenous glucose production. Growth hormone induces insulin resistance, so it's a diabetogenic hormone. If you give it too much hormone for an animal, it will induce diabetes, problems in their glucose homeostasis. And growth hormone also has a very powerful lipolytic action on the white adipose tissue. So this is classical, well described. You can find this in numerous review papers, these metabolic actions of growth hormone. And today, I will talk about how growth hormone can act in the brain, using the brain as an important target to regulate not only metabolic aspects, but also cognitive, behavioral functions too. So I start the question, is the brain a target of growth hormone? Of course, yes. We just saw in the previous presentation. I will, of course, focus in the central nervous system, not in the peripheral nervous system. And one of the things that when we decide to study growth hormone, one of the key doubts we had at that moment, about 10 years ago, was, well, we cannot find a very good description about where the cells that express growth hormone receptor are located in the nervous system. So there were papers showing, oh, there's expression here, there's expression there. Most of them not exactly histological studies. So we could not know exactly which specific cell populations are responsive to growth hormone in the brain. And why was so difficult? Because the expression of growth hormone receptors is normally low in many cells. So it's difficult to detect. Using techniques like in situ hybridization, sometimes you are not sure if that's the background or is a real expression. The same problem happens with other hormones. I have a lot of experience with leptin receptor, and some areas you cannot say clearly that there is expression there because the expression is too low. Another approach, we can use antibodies to label growth hormone receptor. However, those antibodies cannot label properly in terms of histological techniques. Therefore, we came to a different way to visualize the growth hormone responsive cells in the brain. This methodology is exactly the same that people that use to analyze cells in the brain that express prolactin receptors, leptin receptors, all kind of hormones that induces the JAK-STAT pathway. So what we do, basically, we inject a mice with growth hormone, and then after a while, can be, for example, 30 minutes, which is the time for the hormone enter into the brain. There is the growth hormone enters in the brain. We are sure about this, and induce the activation of its receptor. So it's a membrane receptor. When growth hormone binds to this receptor, it will induce the activation of several intracellular pathways. The most classical one is the phosphorylation of STAT5. STAT5 is a transcription factor that goes into the nucleus and mediates important actions of growth hormone in that tissue. So our approach was to label not growth hormone receptor, but cells that express STAT5 phosphorylation after you stimulate the animals with growth hormone. So in these pictures, we can clearly see the difference. On the top, there are animals that receive saline injections. Well, there is expression of pSTAT5 in animals that do not receive growth hormone. Of course, there are. You will see, for example, in this area, because we have in the body systemically hormones that induce pSTAT5. But when we inject growth hormone in those animals, see the difference on the pictures below. So there are many more cells that responded to growth hormone inducing pSTAT5. And those cells certainly express growth hormone receptor, and nowadays we have clear proofs. I will show some proofs that this approach, I would say that's the best way in terms of resolution, because you have a cellular resolution to confirm that the cell expresses growth hormone receptor. So when we did this experiment, it was like a groundbreaking in our lab, because wow, there are so many areas expressing growth hormone receptor in the brain, and we didn't have any clue about that. And when we saw that, we created this idea that is kind of, there is a growth hormone puzzle in the brain. So different areas, each of these pieces represents an area, the name, the abbreviation of that area that contains growth hormone responsive cells. And what we were doing during these 10 years was to induce genetically modified animals to target delete growth hormone receptor specifically in each of these neuronal population. It's like a very hard work. Let's try to see what's going to happen when I delete growth hormone receptor from this area. And then we study the phenotype of the animals. And so on. We were doing this. Right now we have been able to study about 25 different neuronal populations. I will have time to show a couple of them today. So the first two areas that I would like to talk about, and it's nice because we saw in the previous presentation some studies showing results, possibly results about these areas that are the bad nucleus of stria terminalis and a sub-area of the amygdala called the central nucleus of amygdala. These two areas together are known as the extended amygdala because they are kind of brothers. They are very related regions, although they are in different places in the brain. And what's very interesting about this extended amygdala is because there are a lot of growth hormone responsive cells. See in these pictures, when we inject a rat with growth hormone, we see a lot of p-status responsive cells in the bad nucleus of stria terminalis and central amygdala. The same in the rat, compare with saline injected animals. So it's clearly growth hormone receptor. And this area, of course, there is a heterogeneous, there are several types of neurons on this area. And the first thing that we would like to know is which specific neuronal population in this area express growth hormone receptor. And then we did several colocalization studies, and then we discovered that 60% of neurons in these two areas that express the neuropeptide somatostatin, they are responsive to growth hormone. And when we went to the PubMed to discover, well, what is the role of somatostatin neurons in the extended amygdala, we found out that these neurons are extremely linked with neuropsychiatric disorders. So there are a very nice, robust literature showing that these cells are key in the development of depression, anxiety disorders, and other problems. And what growth hormone has, which relationship growth hormone has with these disorders? Well, going to the literature, we easily find evidence that alterations in the growth hormone secretion lead to alterations in the prevalence of neuropsychiatric diseases. This is just an example. This study shows that children and adults with a higher growth hormone deficiency have a higher prevalence of anxiety and depression. So it seems that growth hormone is important to regulate these moods, these disorders in the brain. So what's exactly the function of growth hormone receptor signaling in these somatostatin neurons? And to accomplish this, we used a genetically modified mouse model that allows us to induce the inactivation of growth hormone receptor only in somatostatin expressing cells. So in collaboration with Dr. John Skopchik in Ohio University, who provided these animals have a pair of lots P sites inside the growth hormone receptor gene. Lots P sites are recognized by the Cre enzyme. And when the Cre recognizes a pair of lots P sites, it will delete the region that is between these lots P sites. And in the case of this gene, it will induce the inactivation of this gene. And the interesting thing is that you can induce the expression of this seizure only in somatostatin expressing cells. For this, we needed to cross 2 mouse models. One is the somatostatin 3 mice, and the other is this growth hormone receptor FLOTS animals. So the pups that carry both genes, and of course the FLOTS gene in homozygosity, they will lose the ability to express growth hormone receptor only in somatostatin expressing neurons. And I can prove here. So you can, doing a colocalization study, you no longer see somatostatin neurons co-expressing with p-STAT5 induced by growth hormone. Note that in the amygdala, in the bad nucleus of stria terminalis, we still have growth hormone responsive cells, but no longer in somatostatin expressing cells. We analyze several aspects in these animals. We analyze alterations in metabolism, nothing happens. We analyze alterations in growth hormone secretion, nothing happens. And this is surprising because the literature says that somatostatin neurons are the major neurons that receives the negative feedback of growth hormone to regulate the growth hormone axis. However, when we delete growth hormone receptor from somatostatin neurons, these animals have a normal size, normal growth hormone secretion. We can discuss this later. But the phenotype that called our attention was this. Since the somatostatin neurons are involved in these neuropsychiatric diseases, we analyze several behaviors in these animals. For example, we use the open field test to analyze locomotion. It's normal. But when we saw how much time this animal spends in the center, which indicates anxiety, animals that do not have growth hormone receptor in somatostatin neurons show a reduction. Then, well, there's something here. Let's do another test that evaluates anxiety. And then we use the dark bots test. And these bots will evaluate how many times these animals goes between those compartments, which indicates if the animals has more or less anxiety. And we can see here that these animals show a decreased number of crosses, which indicates these animals are more anxious. They have a higher anxiety. And finally, we use a classical experiment with the elevated plasmase, and we confirm that the animals that do not have growth hormone receptor in somatostatin neurons, it spends less time in the open arms, they move less in the open arms, so clearly these animals have increased anxiety. Well, there are other behaviors that are possible altering these animals, and in the literature say that ghrelin in growth hormone interact in the amygdala to regulate fear memory. So we want to evaluate if the fear memory of the animals are altered. For that, we use a classical fear conditioning study in which we teach the animals to associate a sound with a food shock. So first the animal hears the sound, and then he will receive a food shock. You can imagine that the animal does not like it. And then later, you repeat, and you repeat this a few times. And when you keep repeating this, the animal are learning to associate those two things. And as an expression of fear, these animals spend more time freezing. So freezing is like a behavior that indicates that the animal are feeling fear at that moment. So we can see that the animals learn the task. They learn to associate both things. Then, in the next day, we evaluate those animals in a different place. So the animal could not recognize the environment, but we play the sound. As soon as they heard the sound, what they thought, I will receive a food shock. But they didn't receive. We want to evaluate what? How much time they spend in freezing, and we can see that animals that do not have growth hormone receptor in somatostatin neurons show a less freezing, suggesting that these animals have a reduced fear memory. So very briefly, this paper is published, so if somebody wants more information, can find in the paper. Basically, our results suggest that growth hormone has an anxiolytic effect, which fits quite well with the data of humans, because if you don't have growth hormone, you have higher anxiety, right? So it's an anxiolytic hormone. And when you delete growth hormone receptor from somatostatin neurons, the anxiety increased. So it seems that this anxiety, this effect in anxiety is associated with growth hormone action in somatostatin neurons. And we also saw that growth hormone regulates fear memory by acting these somatostatin neurons in the amygdala. And why this is important? This is important because fear memory is a key feature of post-traumatic stress disorder. And there are also literature in human beings associating the somatotropic access with the development of this post-traumatic stress disorder. Well, let's move to another story. Now I will enter a little bit in our metabolic fields. So I want to talk a little bit about a very recent, it's still unpublished, study where we investigated the growth hormone action in this area called the paraventricular nucleus of the hypothalamus. It's a very important area associated with neuroendocrine control, metabolism, behavior, stress, autonomic nervous system. So see that this area that has this butterfly shape is a very beautiful nucleus. Express a lot of growth hormone responsive cells. And we took advantage of the fact that C1 gene in the brain approach, as I mentioned earlier, we use the C1 mice crossing with the growth hormone receptor flots animals to induce the deletion of growth hormone receptor only in the PVH. Note that the other areas are still responsive to growth hormone. So these animals are normal growth. Just the PVH, the butterfly shape disappear here. So what's the consequences of deleting growth hormone receptor in C1 expressing cells, which targets the paraventricular nucleus of the hypothalamus? Again, we evaluate many aspects, nothing happens. But when we analyze glucose homeostasis, a very striking phenotype caught our attention. So note that animals that do not have growth hormone receptor in C1 cells express lower glycemia, less insulin levels, reduced HOMA-IR, which is an indicator of a better insulin sensitivity. And then we did numerous tests to evaluate glucose homeostasis. For those that are not familiar, this is the glucose tolerance test. We inject glucose in the animals and see how much glucose goes up. Note that the increase in glucose levels is reduced in the knockouts. This test is the insulin tolerance test. We inject insulin, so our expectation is to reduce insulin glucose levels, and this reduction was significantly lower in the knockouts, indicating that these animals are more sensitive to insulin. And then the pyruvate tolerance test, which evaluates how much glucose is produced by the liver. Again, these animals have a lower endogenous glucose production in this test. To confirm that we are talking about changes in insulin sensitivity, we were able to do like a hyperinsulinemic euglycemic clump. So during this test, we infuse insulin, so the glucose levels should go down, but we also infuse glucose, and then we clump it glucose levels. We can see here that glucose levels of the animals started differently, but they were clumped. However, what we measure, how much glucose we infuse. And if you infuse more glucose, it indicates that these animals have a higher insulin sensitivity. That's exactly what we saw. So the deletion of growth hormone receptor from the paraventricular nucleus of the hypothalamus robustly increases their insulin sensitivity. How? It changes glucose uptake? No, glucose uptake is normal. But what changes is the endogenous glucose production. Of course, it's a function of liver. We see that in the basal state, there's a reduction, explaining the lower glycemia. And during the clamp, when we infuse the insulin, insulin suppresses endogenous glucose production. And this suppression is much higher in the knockouts, confirming that these animals have an extremely high sensitivity to insulin in the liver. And we confirm this by analyzing, by using Western blot, the intracellular pathways, insulin injection in the liver, in the body, in the knockouts, will induce a higher phosphorylation of AKT. That's a marker of insulin receptor signaling pathway. So, again, very briefly, this data indicates that the paraventricular nucleus of the hypothalamus is an important area that senses growth hormone levels, and growth hormone receptor expression in this area regulates hepatic insulin sensitivity. How the brain can regulate hepatic insulin sensitivity? We know that there's the sympathetic and parasympathetic nervous system that regulate visceral function. The brain also regulates hormone secretion, so all these effects are able to make the brain control some tissues like the liver. And this is important because, as I told you, growth hormone is a diabetogenic hormone. So now we have the information that at least part of this diabetogenic effect of growth hormone depends on its interactions through these C1 expressing cells. As we are talking about glucose homeostasis, our lab not only studies growth hormone, but we have like a research line about pregnancy. Pregnancy is an amazing model to study metabolism. Pregnant animals develop insulin resistance, increased food intake, increased adiposity. So it's a model similar to diet-induced obesity, but it's physiological. And more importantly, it's transitory. As obesity, it's difficult to treat. So we want to understand if growth hormone acting in the brain could regulate these metabolic aspects, because there are other hormones that act in the maternal brain and do regulate many things, metabolism, behavior, like prolactin, leptin. And one important thing to be considered is that during pregnancy, there's a huge secretion of growth hormone. In humans, the placenta produces growth hormone. In rodents, there is no placental production, but the pituitary gland increases the pulsatile of this hormone. So there are more growth hormone in the circulation of pregnant animals. So to investigate this, we used female mice. And here I would like to show first the results of non-pregnant animals, virgin. And we used in this experiment 2 animal models. One in which we deleted the growth hormone receptor from the entire brain, shown here in blue, and the other that we deleted growth hormone receptor from leptin receptor expressing cells. Well, why delete growth hormone receptor from leptin receptor cells? Leptin receptor cells, especially those in the brain, are major regulators of metabolism. So we want to understand this crosstalk between these 2 hormones. So we see that in virgin animals, this deletion did not cause any significant impact. However, when we analyze the insulin sensitivity during pregnancy, we saw that insulin reduced much more significantly the blood glucose levels of these knockouts compared with the control animals. And the insulin levels are also reduced. Therefore, when you remove the growth hormone action in the brain or in leptin receptor cells, you improve a lot the sensitivity of insulin during pregnancy, suggesting that growth hormone acts in the brain of pregnant animals, inducing insulin resistance. That should be a problem if there are compensatory effects. And we also saw that when we deleted growth hormone from the entire brain, the pregnant animals show reduced food intake only during pregnancy, and also a reduced fat accumulation during pregnancy. Therefore, part of these metabolic adaptations during pregnancy depend on the central action of growth hormone. So growth hormone, like prolactin, leptin, and many other hormones that act in the maternal organism, orchestrates metabolic changes. And this, of course, there is importance in terms of evolution. I will discuss this later. It's not bad. It's good. It's good for the babies. Now, I will talk a little bit about another area that's called the ventromedial nucleus of the hypothalamus. This area has this round-shaped format here. It's an important classical area. In the past, it was called the satiety region of the hypothalamus. And we know that the ventromedial nucleus of the hypothalamus regulate metabolism, regulate glucose homeostasis. Importantly, these neurons are glucose-sensing neurons. So they detect variations in blood glucose levels, and based on these changes, it induces the regulation of our glycemia. It's connected with this circuitry that makes the brain control blood glucose levels properly. So the brain is important to not only for these higher functions, thinking, deciding, moving, feeling, but the brain also regulates how our body works, including the alterations in blood glucose levels. And this area is important for these aspects. So one important thing is that the ventromedial nucleus of the hypothalamus expresses this gene that's called SF1. And in the entire brain, SF1 is only expressed in this area. So again, we can use the same approach. We use the SF1 premise to induce the deletion of growth hormone receptor only in VMH needles. See how beautiful is the deletion. So this area does no longer responsive to growth hormone, and other areas still respond to this hormone. Again, we analyze many things, nothing happens. I will just give this example. We analyze glucose tolerance, we analyze insulin sensitivity through the hyperinsulinemia or glycemic clamp. So these animals have normal glucose levels, normal glucose tolerance, normal insulin sensitivity. However, when we infused insulin in these mice, we saw that the knockouts exhibit lower glycemia compared with control animals. Normally, people in the metabolic field, when they see this result, the first interpretation of these animals have a higher insulin sensitivity. No, but the insulin sensitivity was analyzed for a better method, that's the clamp, and it's normal. Therefore, there should be another explanation. Yes, these animals have a difficulty to recover from the hypoglycemia induced by insulin. So when you infuse insulin, you are causing a drop in blood glucose levels. If you don't prevent it, you can die. So we have mechanisms to prevent this fall. And these animals have defects in this prevention. And I can prove this by infusing another drug that's called 2-deoxy-D-glucose. This drug impairs the ability of the cells to use the glucose. So in the brain, it will make the neurons believe that blood glucose levels is very low because it's competing with glucose in the glucolysis processing. So as the brain thinks that these animals are hypoglycemic, in response, it induces the counter-regulatory response to try to recover blood glucose levels. And we see this huge increase in blood glucose levels that is not caused by 2DG in terms of we are not assessing this kind of glucose in the blood. We are assessing endogenous glucose production by the animals induced by this counter-regulatory response. And again, the animals that do not have a growth hormone receptor in the VMH neurons show a significant reduced response. I will not have time to go into the details. We have more information in the paper, but if we co-infuse 2DG with atropine, that is a parasympathetic blocker, we can preclude this difference. So this result with others that we have in the publication indicates that growth hormone acts in the VMH to induce the counter-regulatory response, and the brain can regulate endogenous glucose production during this situation, probably regulating the parasympathetic nervous system. So, and what's the clinical implications of this? They're important. We know for a long time that growth hormone, that hypoglycemia stimulates growth hormone. So if you infuse insulin and there's this drop in blood glucose levels, you increase a lot growth hormone secretion. Some hospitals still use the insulin test to analyze if a child has defects in growth hormone production. It's not the usual test because of the side effects. Okay, there are other tests that will replace this, but it's clearly a high growth hormone secretion in hypoglycemic situation. And we know in the medical fields that children that have problems in terms of secretion of growth hormone have spontaneous hypoglycemia. That's a side effect of having low growth hormone levels. So you don't have probably this alert signal in the brain to induce the counter-regulatory response and consequently to recover from the hypoglycemia. Now, let's move to this area that's called the arcuate nucleus of the hypothalamus. The arcuate nucleus of the hypothalamus is also a very heterogeneous region. Although it's not big, it's considered the key area that controls the metabolism in the entire body. So neurons in this area regulate our food intake, our energy expenditure, and many other metabolic endocrine aspects. So we were able to delete growth hormone receptor from different neuronal populations in this nucleus. However, I will focus my presentation in this specific group of cells shown here in green that's called the AGRP neurons. Why studying the AGRP neurons? AGRP neurons are the master regulators of our hunger. If you are feeling hunger, you are feeling hunger because AGRP neurons are active. And the release of neurotransmitters from these neurons is one of the masters inducers of this feeling in our conscious feeling. And not only AGRP neurons stimulate hunger, but the activation reduce energy expenditure. They co-express neuropeptide Y, that's also an orexigenic neuropeptide. And importantly, several hormones regulate metabolism through AGRP neurons. So when if you heard about this hormone leptin is critical hormone to regulate our metabolism, which is the main neuronal population that is involved in this metabolic regulation of leptin, AGRP neurons. And so on, with insulin, ghrelin, and so on. Therefore, AGRP neurons express growth hormone receptor. Oh yes, and we were not the first to discover this. 30 years ago, people already showed that 95% of AGRP neurons co-express growth hormone receptor. And using the p-STAT5 approach, we confirm practically all AGRP neurons are responsive to growth hormone. This is amazing. And when we infuse animal with growth hormone, like a pharmacological injection, and if you recollect the hypothalamus, what we saw? We saw an increase in AGRP and NPY expression. This is an indication that growth hormone is activating these cells because it's inducing the transcription of these neuropeptides that are co-expressed in these cells. And to confirm this, we perform whole cell patch clamp recordings, and we can clearly see here that the application of growth hormone in the brain sections induce this beautiful depolarization, confirming that AGP neurons are activated by growth hormone. And that can explain these results. If we put in a cannula infused growth hormone in the ventricles, it's an intracerebral ventricular injection of growth hormone, it's a central injection, we see that growth hormone stimulates food intake and reduces energy expenditure. And this is exactly what happens when you induce the activation of AGP neurons. So, growth hormone probably uses AGP neurons to induce those responses. And here I also would like to call the attention for evolution. I think evolution is everything. We need always to imagine our studies put in the context of evolution. Why growth hormone would stimulate food intake? Well, growth hormone, as the name says, stimulates growth. To growth, you need energy. So evolution probably coupled these two tests to make the animal, when they secrete more growth hormone, and when we secrete more growth hormone, when we are childs, when we are in the pubertal period, so when we are growing, and as we saw children and adolescents, they eat a lot. So probably growth hormone is inducing this higher feeding behavior, and this is important for growth. So I think evolution was smart to link these two functions. Well, using our usual approach, deleting growth hormone receptor from AGP neurons, so we were able to manage this, we were analyzing possible metabolic alterations in these animals. Initially, we did not see anything. These animals have a normal body weight, normal body composition, normal food intake, energy expenditure, anything different. Well, let's challenge those animals. And how we challenge AGP neurons? Putting the animals in fasting states, because fasting induces the activation of these neurons. And the activation of these neurons causes the hunger feeling that a fasted animal feels, right? So when we put these animals in fasting, we were able to see a significant reduction in the number of AGP neurons expressing CFOs. CFOs, as many of you know, are a marker of neuronal activation. So it seems that less AGP neurons are active when you don't have growth hormone receptor in these cells during the fasting state. And to confirm this, we use a different protocol. Instead of putting an animal in fasting, we food restricted the animals. For 3 days, the animals received 40% of their usual food intake. So they lose weight, they were hunger, and then when we analyze the gene expression in the hypothalamus, we see in control animals, see in white here the control animals, increased NPY and increased AGP expression. So this increased activity of these neurons are inducing the hunger feeling, etc. And animals that do not have growth hormone receptor in AGP neurons show a prevention in this activation. So every time that you are food restrict, you have the activation of AGP neurons. And it seems that this activation requires growth hormone receptor signaling in these cells. I will not have time to put some information, but chronic food restriction, not acute, chronic food restriction increases blood growth hormone levels. So growth hormone is secreted during chronic food restriction. And every time that you induce the activation of AGP neurons, you have the hunger feeling, you have a reduction in energy expenditure. How the body can regulate energy expenditure? Suppressing process that will not make you die, but it's a way to save energy at least for a moment. So the thyroid axis is suppressed, the reproduction is suppressed, the thermogenesis is suppressed. So we evaluate all these aspects in our animals. Note that during food restriction, control animals have a reduction in T4 levels, a marker of thyroid axis, a reduction in testosterone levels, a marker of reproductive axis, an increase in corticosterone levels, during food restriction, and reduce expression of uncoupling protein 1 in brown adipose tissue, which is a marker of thermogenesis. Why the control animals showed these neuroendocrine changes? Because they need to save energy. If you are not finding food, you will die. That's what happens in nature. So one way to postpone this final consequence is decrease energy expenditure. If you decrease energy expenditure, you can have a little bit more time to find food. And control animals make this beautiful response. But animals without growth hormone receptor in EGP neurons does not reduce T4, does not reduce testosterone. The corticosterone levels does not go up, and the suppression of the uncoupling protein receptor, uncoupling protein 1, is also prevented. And what's the final consequences of all this? While control animals save energy during food restriction, so in the basal state is 100%. 1 day of food restriction decreases in 10% the energy expenditure of control animals. And each day of food restriction, the energy expenditure is dropping. And what happens in the knockouts? These animals that in the basal state have a normal energy expenditure, they were able to maintain an elevated energy expenditure during these periods, causing, of course, a higher weight loss, and this weight loss was predominantly loss of fat mass. Therefore, summarizing what I just told you, what's our hypothesis? Our hypothesis is that like leptin, the fall in leptin levels is a brain signal that, oh wow, we are not finding food. But not only leptin does this. Growth hormone during food restriction, especially chronic starvation, growth hormone secretion is elevated. It will induce the activation of growth hormone AGP neurons. The increase in release in these neurotransmitters will not only induce hunger, but also energy-saving adaptation. So growth hormone can act as a starvation signal to stimulate hunger and to prevent, and to make the animal run in an economic way, saving energy. When you see in terms of evolution, this adaptation is important because you have more time to find food before dying of starvation. But in the context of obesity treatments, this is bad because every time we lose weight, this response happens and we save energy. Saving energy, it's going to be more difficult to lose weight, and it's going to be more easy to recover the weight. Okay? So we think that our findings can somehow help in the future development of ways to prevent these adaptations that save energy. And to finish my presentation, I will just also quickly say something about the growth hormone receptor expression in this lateral hypothalamic area. So the lateral hypothalamic area is a very important structure which studies sleep, arousal, metabolism. Many aspects are regulated by the lateral motivation, the dopamine secretion. So lateral hypothalamic area is a key structure involved in many of these functions. And as we saw in other areas, lateral hypothalamic area expresses a lot of growth hormone receptor cells. We see the responsiveness to p-STAT5 in this area. And when we colocalize with different markers of the lateral hypothalamic area, interestingly, 70% of orexin neurons in the lateral hypothalamic areas co-express growth hormone receptor. See, what's the role for orexin? Orexin not only stimulates food intake, as the name says, but it's a key hormone for the regulation of arousal, motivation. The lack of orexin or orexin receptors causes narcolepsy. That's the molecular cause of narcolepsy. So it's really interesting to why growth hormone is acting these neurons involved in sleepy or arousal. Well, we do not have a crystalline able to manage this. And then we use a different approach. we put, using stereotactic surgeries, we were able to inject a virus that induces the Cre expression in the lateral hypothalamic area. And as we injected this Cre virus in growth hormone receptor flox animals, we were able to delete growth hormone receptor only in the lateral hypothalamic area, preserving other nuclei that are also responsive to growth hormone. So what happens when we delete growth hormone receptor from the lateral hypothalamic area? We analyze metabolism because the lateral hypothalamic area is involved with dysfunction. Nothing happens. We then decide to put these animals in food restriction because we have those previous work showing that previous restrictions is nice, etc. And well, nothing happened here. We see normal weight loss, a normal energy expenditure, a normal substrate oxidation. But when we analyze the ambulatory activity, the voluntary ambulatory activity of these animals, note, this is very interesting. Control animals respond to food restriction by increasing their locomotor activity. Why? It's like us. If we are hungry, you cannot be quiet. You are annoyed. So you don't want to sit. You are feeling hungry, this bad feeling. So the animals are the same. So every time that the animals are feeling hunger, they move more in the cage. Evolutionary, we think that it's a way to motivate the animals for food-seeking behavior. They want to explore the environment to find food. Imagine if you are in the wilderness, if you are hungry, if you stay quiet, the food will not fall in your head. You need to go and find food. So this behavior, it seems really important in evolution. And animals that do not have growth hormone receptor in the lateral hypothalamic area, this increased activity, it completely disappeared. And to prove that is the food-seeking behavior, we tested those animals in this food-seeking behavior test. We hide a food pellet, and the animals need to find this food pellet in the cage. If they are in ad libitum states, fed states, these animals take a long time to find the pellet. Well, I don't care, I'm not hungry. But if the animals are hungry, they quickly find the food pellet. They smell, they start to search in the cage until they quickly discover. And if you don't have a growth hormone receptor in the lateral hypothalamic area, this response is also significantly attenuated. We analyzed the activity of orexin neurons. So when the animals are starving, they have a higher activation of orexin neurons, and the animals that do not have growth hormone receptor, this activation analyzed through C4 expression disappear. So summarizing these last studies, what I would like which message I would like to give to you? Well, we think that growth hormone is extremely important to regulate homeostasis in specific situations. One good example is starvation. See the metabolic classical effects of growth hormone. During starvation, as I told you, there is a high secretion of growth hormone. Growth hormone acts in the white adipose tissue, induces lipolysis, and it is providing nutrients for the cells. Fatty acids, glycerol, and this is extremely important, not only for the cells that need those nutrients, but also for substrates for gluconeogenesis. So it is sparing the use of glucose, because in starvation, glucose in the body is precious. Growth hormone acts in the muscle, induces insulin resistance. Well, this is bad. No. If you are in starvation, insulin resistance is good because it will decrease the glucose uptake and make the muscle do not use the precious glucose. Leave the glucose for the cells that need, for example, the brain. And growth hormone acts in the liver and stimulates gluconeogenesis. So it maintains blood glucose levels and avoids hypoglycemia. Finally, and this is our contribution to this field, we saw that growth hormone can act in the brain during starvation and stimulates hunger, save energy, and in this last study, it stimulates the search for food, the motivation to search for food. Everything, of course, is helping to make the animal have more chances to survive in situations of metabolic stress. Starvation is a situation of metabolic stress. Pregnancy, yes, it is too, because the organism of the mother needs to manage more energy than they were used. Exercise, I did not have time to go through our results in terms of exercise, or hypoglycemia. See that growth hormone, either in their peripheral actions or the central actions that I just showed to you, it's extremely important to maintain homeostasis, to avoid that you die during these situations of metabolic stress. Oh, what about anxiety? There's nothing, no relationship? Of course, no, there is relationship. In my opinion, growth hormone, by acting as an anxiolytic hormone, will make the animal express behaviors to go out of this situation. Imagine if the animals got anxious during a starvation, so the animal will not risk themselves to find food. So during situations of metabolic stress that your life is in danger, perhaps decreased anxiety is a good adaptation. And what we saw is that growth hormone seems to be a signal, a tool that acts in different neuronal populations, coordinating these responses to make us survive in the situations that put our life in threatening. Well, in finishing my presentation, I just would like to show that this is, for me, beautiful. Why so many cells in the brain express growth hormone receptor? The answer is probably because each neuronal population has its specific function. One neuron regulates hunger, other food-seeking behavior, other the control for blood glucose levels. And by presenting growth hormone receptor in all these neuronal populations, one single molecule, one single hormone can act in these different pathways in the brain and coordinate numerous functions, metabolic, behavioral, etc. And all these responses, of course, they will have the same final objective, that is probably make you survive, especially in situations of metabolic stress. I would like to thank people in my lab that work in these studies, my former, my present studies, our collaborators, funding agency. Also, I would like a special thank you for all that are hearing my talk and for the very kind invitation to be here. Very gentle people. Thank you so much.
Thank you for a very exciting and complete frame on growth hormone actions. You mentioned deleting the growth hormone receptor in whole brain, and you focus on pregnancy. Yet, in the view of all the different things that growth hormone does in the brain, wouldn't you expect to have the phenotype of all these actions in that model? Why focus only on pregnancy? And how did you delete it in the brain? Okay,
thank you very much, Carmen, for the questions. I have several answers for these questions. One is that, of course, it's pretty nice to see those models, so they work fantastically. They allow us to do some very specific questions, but they are not perfect. And one model that is not perfect is the growth hormone receptor deletion in the entire brain using the nesting tree promoter. So this mouse model has some problems. And these problems made us avoiding using this animal in multiple experiments. And that's why sometimes the phenotype of these animals cannot replicate all the other phenotypes are not in these animals. In some aspects, yes, for example, the food-seeking behavior, perfect well. But some others not, but because this animal has some problems, issues regarding the construction of the genetic modification. This is one thing. Regarding, we used this model, the brain-specific deletion, in other experiments too. So I did not have time to show all these results, but just a quick example. In the last results I show about food-seeking behavior. To make the point that it's the orexin neurons involved in this, we use the animals that we induce the deletion in the entire body or in leptin receptor expressing cells, because the leptin receptor expressing cells in the lateral hypothalamic area is another neuronal population that also are responsive to growth hormone. And when we delete growth hormone receptor from leptin receptor expressing cells, nothing happened in terms of food-seeking behavior, but when we use the nesting, yes. So that was one of our first studies published in 2019. And right now, we avoid to use these whole brain deletions, and we are trying more very specific neuronal populations targets.
Right, because all of these actions were not apparent when you removed the growth hormone receptor from the whole animal. In fact, it was claimed that they were just perfectly normal, just tiny, just dwarfs. So all of these, the animals survived perfectly, they developed perfectly, they just had a change in size. So would this mean that these are very subtle effects that need other interactions, other hormone interactions, other mediators within the brain to really show the influence of growth hormone? By itself, you don't see them, but you need the interaction with a complex endocrine,
that's really nice
physiological environment
for the students, because knockout animals have the problem that the whole body knockout animals that they are. We know this for many examples that they have compensatory effects. So as the deletion is higher, for example, the whole body, that's a dwarf animal. We do have those animals in the lab. These animals, probably the organism, since like a germline absence of growth hormone receptor, they will adapt. And the brain is plastic. That's one of the things of. So if you delete growth hormone receptor in specific neuronal populations, we believe that we have less chances to have this plasticity, this adaptation, than when you have like the whole body knockout animals. That's one possible explanation. The second explanation that not all the behaviors or the metabolic studies that we did indeed were reproducing the knockout. So that's perhaps I would say that some of those behaviors may be present in these animals, but nobody studied those. But I think the ability of the animal, especially the brain, to adapt to a very strong deletion is clearly present. Just one example that I think illustrates quite well this. In the hypothalamus, the AGRP neurons are the key mediators of hunger. If you kill these neurons in newborn animals, the animal will live practically normally. But if you kill AGRP neurons in adult animals, these animals will die of starvation. So it's a good example that the network managed to work without that specific signal. That's our belief. But of course, there are many studies to be done.
Thank you.
Thank you.
Hi. Thank you very much for your talk. I am thinking, what about in the contrary, in the metabolic syndrome? What happened with thyroid hormones and growth hormones and other things in the brain with the opposite? That's
an excellent question. We are investigating more about this now. Obesity or diabetes reduces growth hormone secretion. It induces like early somatopause. So we know that during aging, growth hormone secretion will decline. And if you are obese or diabetic, the growth hormone will go down. We don't know exactly why. Which drives this? That's what we are studying. We think that it's insulin, possibly free fat acids. There are many possible explanations. So, but the fact is that an obese individual is kind of a mild growth hormone deficient individual because of this. So, as we saw in the previous talk, growth hormone is important for neuroregeneration or prevention. That can be an issue about obesity or diabetes. These responses, they are reduced too. If you do like a responsive test in an obese individual, they will secrete less growth hormone. So probably they have like impairment in all these aspects that depends the growth hormone. So it's part of the obesity. Some people propose the possibility of replace growth hormone in obesity. One of the possibilities is to use the anabolic effect of growth hormone. You gain muscle, you lose fat. But the problem is that growth hormone makes the individual lose weight. Yes, if you increase the dose, you will lose a lot of fat mass. Acromegalic animals, you don't see fat. If you try to dissect it, it's really hard. These animals kind of almost nothing of fat. So it's everybody that would like something. But what's the problem? You gain a package of problems associated with the excess of growth hormone. Excess of growth hormone will make you diabetic. So that's the main side effect. And we have some evidence that in situations that chronic over-secretion, you have neuroinflammation. And neuroinflammation is involved with neurodegenerative diseases, etc. So perhaps evolution reduced the growth hormone to prevent this effect in the brain, but you are losing many of these effects. So maybe it's kind of a comorbidity of obesity, we could say. As obesity have different alterations in hormone secretion, right? So if you see the hormone profile of obese individual, they will have several changes. One is low growth hormone secretion, and you gain the whole package about this.
Yeah, it's almost the all people when add the growth hormone increase the prostate and mammary cancer.
Exactly.
Huh? Mm-hmm.
Yes, the literature about growth hormone cancer is controversial, but yes, it's a proliferative. You would risk yourself to have something that not only induces obesity, but can increase cell proliferation. So we don't know actually. So it's better not to risk it. So that's the problem of those clinical trials. But they improve you some stuff. One thing in terms of, this is amazing. I did not find this area. We are searching for this area. But when we talk to physicians that take care of patients that are growth hormone deficient, children or adults, one thing that's common, they say that these individuals have low energy. So they're it's in their heads, not low energy. They simply, I don't want to do, I'm too tired, and etc. So it seems that the motivation system are kind of shut down. And when you replace it, you improve a lot of this. It's the main factor that makes the people that take the growth hormone replacements more happy, more, the quality of life increases, especially because of this feeling that, now I feel more energy, I want to do more stuff, and etc. So you see, there are so many effects of growth hormone that we don't know. But I think the point is, there's a balance that's important, this balance. And the second thing is that the brain's important target. We ignore this for many times. So either in the peripheral tissues, we saw the results of the Carlos lab, and etc., but also in the central nervous system. We should put the central nervous system, the peripheral central nervous system, as an important target of growth hormone. So important as the liver, as the muscle, as the pancreas, or this classical. And we don't see in the textbooks. So when we teach physiology, I teach physiology for my students, now I tell about them. But when I asked them to read a book, there is nothing in the book. So we need to change this. We need to put the brain as an important target of the growth hormone.
Thank you very much.
Thank you. I wanted to ask, related to the last question regarding the thyroid disease, what can it happen with the hormonal response that might kind of develop as a chain reaction with sexual hormones, especially in women, since we are the most affected by any hormonal disbalance and can interfere with the fat metabolism. It can be used, this approach, with the growth hormone.
Well, we normally in our studies we try to use both sexes to analyze sex dimorphism. Sometimes we find, sometimes sex dimorphism, sometimes we saw the same phenotype. Growth hormone acts is extremely relevant in terms of there's a sex difference. The pattern of secretion is different in males and females. And the pattern of secretion makes a huge difference in the liver function. People that's not in the field don't imagine it, but our liver, it's a very sexually dimorphic organ. Hundreds of genes have a different expression between males and females. The main factor that induces this sexual dimorphism is growth hormone. And this makes, for example, men respond different to women a certain type of drug. That's why clinical trials should put women. We know that in the past, the literature, many people are just using males. It's easy. There is no the astrocyte, etc. But it's important. So there is this sex difference that have many implications in the body. And in terms of how sex hormones interact with growth hormone, there is a paper published 4 years ago from my previous supervisor, where they inactivate estrogen receptor alpha from GHRH neurons. So GHRH neurons are neurons that produce the peptide called growth hormone releasing hormone. That is the main stimulator of pituitary growth hormone secretion. And when estrogen receptor was removed from the cells, growth hormone secretion were decreased. So it seems that estrogens have a positive effect in growth hormone secretion, regulating, and we don't know exactly how is the interaction of in terms of functions. As I told you, some of our studies show similar phenotypes, others not. So it's kind of depending on the aspect you analyze. I don't know if I answered your question.
Yes.
Okay, thank you.
Thank you for your talk. That's a nice talk.
Thank you.
About the behavioral results that you show us, you show that this GRH ablation is related to fear. So in the last task that you test, which is the conditioning, I want to know if it's related to learning and memory processes, because they reduce anxiety and fear, but fear is related to all the models that we use in learning and memory, so maybe it can be involved in that, especially those neurons in CA.
Yes, no, it's a very good question. So, well, which evidence we have? So, both knockouts and the controls learn the task equally well. So there's no difference. So the acquisition of the response of expressing fear during the fear conditioning was similar. So no difference in all parameters that we evaluate. We analyze the other aspects regarding memory in those animals. For example, contextual memory. It's normal. So these animals does not have all kind of memory problems. But these animals seems to have like a problem, impaired fear memory, exactly what's going on. If it's the acquisition or not, we did not explore this. But this same aspect was explored by other researchers. So there are 2 papers, one published in 2014 and 16, very nice papers published in very good journals showing that ghrelin, probably many of you heard about this hormone, but if you write ghrelin with GH, that starts as ghrelin releasing hormone. That's why this word came up. So it's the hormone that induces growth hormone secretion. Ghrelin is a powerful growth hormone secretagogue. And this induction of growth hormone secretion is not only in the pituitary glands, but also in extra-pituitary. And these previous papers show that ghrelin receptors are expressed in the basolateral amygdala, not in the central, but in the basolateral. And the activation of ghrelin over there induces the expression of local growth hormone. And what we think is that this growth hormone, basolateral amygdala, does not have growth hormone receptors. They have ghrelin receptors. But probably there is this local production. We think that by diffusion, it reaches the central nucleus of amygdala, and that can be the association about this fear memory. In that paper, they show that the dendrites changes related with fear memory are implicated, are altered, and so they give you much more this kind of information. In our paper, we want to identify the neurons that express growth hormone receptors to receive this signal. But this signal can be from the pituitary gland, but can be from this local circuit. So it seems specific for fear memory, not, and the acquisition was okay, but we did not explore the details exactly what's changed. But this previous paper showed that it is the synapses change, the dendrites changes, so they are important structural changes that are growth hormone dependent in these areas.
Thank you.
Thank you very much. That was a very exciting talk, and it made me think how evolutionary these hormones evolve to regulate in a very fine way how the system works, how the homeostasis works, the feeding, the food-seeking behavior, and that nowadays with this overeating, having food all the time, we are really making this evolution not working well anymore. So thinking, how can we overcome this? Well, there are many ways, of course, but how about food restriction, these new protocols on food restriction or time-restricted feeding? Do they involve increases in growth hormone levels? Okay,
I'm not sure about this specific foods time-restricted feeding protocols. We know that there is this a lot of, especially in humans, but in animal models, that you allow the animals to eat for specific moments, and this can alter many metabolic aspects. It's kind of a diet that people say that can be advantageous. This protocol, I really don't know if it changes. In terms of food restriction, it's really interesting because in the literature, it's kind of, some papers show that food restriction increases, other not. So we tested this detailed. So what we saw that acutely, for example, if you give like overnight fast, an overnight fast for mice is not the same overnight fast for us. Overnight fast for us is physiological. You will wake up, okay. But for mice, they need to eat during the diet cycle, and they need to eat frequently, so they are kind of extremely hungry. They lose like 10-15% of their body weight during this period. This protocol completely blocks growth hormone secretion, as it blocks several pituitary hormone secretion. It's interesting. Several hormones are kind of not say shut down, but suppressed. But if you put in an animal in a protocol, what we call food restriction, we give you a certain amount of animal to make the animal lose weight. So the animal will lose weight. when they reach like a very low percentage of fat, body fat, then you analyze the growth hormone secretion. In that situation, growth hormone secretion is increased, but not like in normal situations, because what's our normal growth hormone secretion? It's pulses. There are a huge pulse and an impulse interval with low secretion, a huge pulse, and so on. And during food restriction, there are pulses, but there are an increase in the basal secretion. So the pulses always started in a certain level. And what's the implication of this? I will not talk about the brain. I will talk about nutrition, physiological responses in the body. The liver will turn off the response to growth hormone. And this is important because during this starvation protocol, what's the reason to secrete IGF-1? IGF-1 is an anabolic hormone. So you shut down IGF-1, and you have high growth hormone secretion. And the anabolic effects of IGF-1 disappear, and we have the growth hormone secretion probably doing its metabolic effects, for example, causing insulin resistance, acting the brain, and this kind of stuff. And that's what we found like in people that are, undernourished people. So people that are really starved, exactly this. They have what's called growth hormone resistance. Growth hormone resistance in the liver develop because the pattern of growth hormone secretion develops this more basal secretion, more tonic secretion, less pulsatil secretion. How the brain processes these two signals, I don't know. We just put the animals in the protocols and see the result. But for some cells, they still respond to this signal. For other cells, for example, in the liver, they completely shut down. So it's complex. It's good because we will have many years of research. Thank you very much.
Thank you.
Thank you.